All right, welcome everyone. So today's lecture is on RNA biology, RNA transcripts, and RNA folding. So here we are in module 2. This is the second lecture of module 2. In the first lecture, we talked about expression analysis and how to use gene expression levels for clustering, classification. We saw k-means, we saw hierarchical clustering, and we also saw Bayesian inference more generally. Today, we're going to be talking about RNA biology. And then the next two lectures are going to be about epigenomics and the three-dimensional genome. So here we are in module 2. Today, we're going to be talking, number 1, about some biology. So basically why RNA is so incredibly awesomely cool, and then how to infer transcripts from sequencing reads, and then how to fold RNA and understand the various functions that it plays. So we talked a lot about evolution in sort of motivating our we talked a lot about evolution in motivating our comparative genomics, our sequence alignment, our blasting, and all of these models. And we're going to talk a lot more about comparative genomics. But the usual picture that I show is, oh, look at all these lovely little species that you know about. And then there's like a branch going off here. So the question is, what actually happened before the first common ancestor of all life on Earth today? So the quick summary is that life as we know it today is amazingly complex and totally awesomely cool. And I can go on for hours and hours and hours about how awesomely cool life and sort of molecular biology is. And if you look at the various players that are working together, they line up together inside the cell in something that we know as the central dogma of biology. And the central dogma of biology is DNA makes RNA makes protein. So that's sort of, the 3 steps. If you think about those 3 steps, RNA is basically in this DNA makes RNA makes protein view of the modern molecular biology world. RNA is kind of boring. It's like the messenger. DNA is like the awesome double helix, this beautiful molecule. And RNA is the intermediate, and proteins are these awesome sort of effectors that do all kinds of stuff. But RNA doesn't just play the messenger role. That's mRNA. There's also rRNA, ribosomal RNA. And ribosomal RNA is actually the agent that will then, translate mRNAs into protein. mRNA and rRNA are only 2 types of RNA. There's also tRNA, which is basically this transfer RNA, this adapter molecule that was hypothesized well before it was discovered to play the role of an adapter, having on one side this DNA RNA base pairing, Watson-Crick, hybridization that we all know and love, and on the other side having a adapter that allows incorporation of different amino acids to create the proteins that get made out of this messenger RNA. But RNA is also involved in splicing, in splicing associated RNAs, and RNA is also involved in post-transcriptional regulation of messenger RNA in repressing it. RNA is also involved in scaffolding and sort of putting together chromatin in all kinds of ways. So RNA plays many, many roles beyond this sort of temporary copy and boring information we have today. So again, just to recap, DNA serves today the role of information storage and cross-generation transmission. DNA, you don't get it out of the cell that much. You don't do much with it. You kind of use it, safely in a little box so that you can pass on this beautiful treasure to the next generation. Protein, oh, that's messy. It sort of carries out all cellular functions. But as we saw in the previous slide, protein is not the only one that can carry out these sort of complex structures and folding. RNA can also carry out enzymatic reactions. So ribosomal RNA can actually carry out the enzymatic reaction of translating RNA into protein. So RNA can, of course, transmit information, copying the RNA in the DNA into a temporary copy, and then folding that copy and translating into protein. But RNA can also, base pair with itself and transmit information again. But RNA can also store information. So I don't know if you guys have heard about, there's this virus going around, COVID-19. So it's an RNA virus. It doesn't bother with DNA. It is transmitting its genome as RNA. So the SARS-CoV-2 genome is 29,300 something nucleotides of RNA. So RNA is actually the information storage for some organisms, including this deadly thing that has us all stuck at home. So no, there's plenty of DNA viruses and there's plenty of RNA viruses. So not all viruses are RNA. So we're basically seeing this complexity, this diversity of RNA as playing, all three roles of RNA, DNA, protein, but it also plays many, many additional roles. So basically, as I mentioned before, RNA is the ribosome itself that's made out of RNA. The transfer RNA is made out of RNA. And then there's a gazillion different additional functions of RNA. Like there's primer RNAs and telomerase RNAs that are used in DNA replication. When you get to the end of your chromosomes, you don't have any place to start copying from. And that's where telomerase RNA is sort of laying out the foundation for more DNA to be formed. In transcription itself, as I mentioned, there's like a lot of splicing associated factors, but there's also SNO RNAs, mononuclear RNAs. There's large non-coding RNAs that are coating the X chromosome to sort of repress it, like exist RNA. And there's, a diversity of long non-coding RNA functions. There's microRNAs and small interfering RNAs that are involved in degradation by hybridizing to the mRNA and then leading to degradation. And of course, they're also involved in translation regulation. So basically, incorporation into DROSHA and DICER are basically leading to microRNAs playing all kinds of roles in regulating mRNA. We recently discovered in collaboration with collaborators over at the Hannon Lab at Cold Spring Harbor, a new class of pi RNAs. So these are the guardians of the genome. They're basically sort of repressing repetitive elements to avoid the genome getting damaged. And mRNA, basically mRNAs are sort of the boring part. RNAs play tons of additional roles. So here's just one large view of all of the roles that RNAs play in transcription, in translation, in post-transcriptional repression, in processing, in splicing. I mentioned the rRNAs. They're also processing rRNA methylation and pseudouridylation. They're leading to cleavage, instability, or less efficient translation. Again, processing, post-transcriptional regulation. So lots of diverse functions of mRNAs. So here's some examples. This is what I like to call the most awesome RNA machine ever built. So very early in evolution, we got stuck with this ribosome. As soon as RNAs were complex enough to make this translation into protein, that structure of RNA basically was passed on, and all of the living organisms that had it were suddenly able to have a much larger diversity of functions, and therefore all of the other organisms that didn't have the ribosome effectively died. So basically every species today has a ribosome that allows it to translate, and they're all very similar to each other. And the structure of the ribosome was solved a few years ago, and it was truly a breakthrough in showing that the catalytic core is entirely made of RNA. Yes, there's a lot of proteins that are playing auxiliary roles, and you can see them here in color. But in gray and this light cyan, you basically have only RNA carrying out all of the catalytic reactions of the ribosome. And if you look at how that is possible, well, it's because RNA can be single-stranded, as in a messenger RNA, or it can be double-stranded and folding, not just with a very, very long and boring strand that has exactly the same information as in the DNA, but it can base pair with itself. So if you start here at the 5' end of the ribosome, you basically loop around, and this region is complementary. So it will form, partial double helix. And then it'll loop around to this region here, which basically base pairs with itself all the way down here to form another stem. And then over here with itself, and so on and so forth. So RNA forms an incredible diversity of structures through this very, very simple rule of base pair with myself wherever I can find a match, and eventually fold in 3 dimensions, and eventually get all kinds of modifications that allow for additional long-range interactions and loops and so on and so forth. But basically, the beauty of it is that the ribosome catalytic core is made entirely of RNA, and it's effectively the world's most awesome ribozyme. And even the peptide bond formation is actually catalyzed by an adenine in the ribosomal RNA. So basically, even that last part of sort of creating this is actually done by an RNA. And the protein components only play an auxiliary and structural outside role. If you look at splicing, so splicing RNA is at the core of this DNA to protein path. So you basically have, this ability of mRNA to basically undergo splicing to excise away these introns and only maintain the exons. And this ability is actually carried out in great part by splicing, small nuclear RNA. And then these small RNAs are used in both recognition, base pairing, and in catalysis. So they recognize splice sites and they interact with each other and with other proteins. RNA can also replicate itself. So this is actually a self-replicating RNA. This is a ribozyme that acts as an RNA-dependent RNA polymerase. It's a chimeric construct of a natural ligase ribozyme with an in vitro selected template binding domain, which can replicate at least one turn of an RNA helix. So you can basically sort of imagine how RNA may have actually done all of the molecular functions of the cell, without DNA and protein. So both DNA and RNA are made of 4 bases. Why RNA can be so flexible? So I'll get to the structure of RNA and why it's so flexible. But one of the reasons is that instead of having a T, it has a U. And this U allows it to sort of, fold more easily, but it also allows it to generate GU base pairs rather than just A T base pairs. So basically T can only base pair with A, but U, which replaces T, can base pair with either A or with G, creating more versatility. And Pablo responds because it's a single strand, DNA is 2 strands bound to each other, so they tend to fold and stuff. Yes. So basically, there's 2 reasons, of course. Number 1 is that single stranded, and number 2, it has this additional flexible RNA. So as I mentioned earlier, RNA can mediate enzymatic activities, so it can actually catalyze reactions without getting used up itself. So, some very cool examples are self-cleaving ribozymes, the pre-tRNA cleavage of an RNA-P. So self-splicing of group 1 introns that basically just catalyze their own splicing reaction, capping reaction similar to the branching reaction of group 2 introns, self-splicing of group 1 introns using this branching reaction. The branch side is sort of the middle side. There's a donor, there's the acceptor, and the branch side is sort of what leads to that sort of folding. And the hydrolytic self-splicing of group 2 introns. Again, all of these are ribosomal, RNA machineries. So here's the self-splicing introns. You basically can sort of design. And I mean, this is just so awesomely cool as an engineer to basically think, how can you design these folds by simply typing in a sequence into your computer, synthesizing that RNA molecule, and then watching it fold onto itself. And you can be like, oh, I can make another fold here. Let me change that letter to be complementary here, and so on and so forth. So using this, you can basically end up with these catalyzed reactions by effectively designing the structure based on these folds and then typing in the sequence that matches those folds. So speaking of that, you can basically, for example, design an RNA that will have complementarity here and complementarity there. And you could also make that part complementary to this part here, so that in certain conditions, it switches between one and the other conformation. Well, guess what? If you can imagine it, nature has already built it. So nature has actually built ribose switches. These are RNA structures that undergo self-regulatory conformational change. So if one part interacts with, something else, then you basically have that sensor for that whatever something else is, whether it's a small molecule, metabolite, and so on and so forth. You can basically switch between two different conformational structures, leading to, for example, the revelation of a start codon. So basically here, the start codon is hidden. There's a terminator there. You can't translate through this because, you're in the off state. And then as soon as you sense a particular molecule, that molecule can now switch the conformational state of this RNA and basically expose the translation start. And then you can launch RNA translation without any kind of other factors intervening. So the message that I'm trying to get across with you guys is that RNA, in fact, precedes all of the other machinery that we are used to today. So basically, in today's world, we have DNA mix RNA mix protein, and that's our current potential dogma. But in the RNA world, we basically had RNA mix RNA. So basically, RNA was the sort of agent that carries the information. RNA was the transfer molecule that sort of carries the messengers through. But RNA was also the agent that sort of carried out all the reactions. So basically, all of these, and of course, it wasn't this way because RNA could just very happily transmit information at the same time that it's doing self-modifications and self-folding and self-splicing and so on and so forth. So, and self-replication. So the current thinking ever since the 60s is that prior to today's world, very, very early on, that little blue branch that I showed you on the previous slide, lies an RNA world where everything was RNA. And eventually, of course, the question is, how did we get to that RNA world? Basically, there's many, many theories. Again, huge debates and very, lots of uncertainty. But one idea is to get to this RNA world, we basically have some pre-life where clay minerals can basically guide the synthesis of single RNA strands. Basically, nucleic acids are thought to have existed and be spontaneously created under the conditions of early Earth. They were thought to eventually detach, fold, and catalyze, potentially reactions through this. And you could also have either self-replication of RNA or cross-replication, where one enzyme, one RNA molecule, like A favors B, favors C, which favors D, which favors A again. And then you effectively have this reinforcement where I can make small modifications that will then be selected and improved upon, eventually launching RNA world and self-replication. And the moment you sort of enable self-replication, life takes over, because it can, in parallel, explore gazillions of possibilities simultaneously and sort of get better at doing all of the functions that it can do, including self-replication. So that's sort of the early creation of the RNA world. And then once you have this RNA world, how do you get to today's world? Well, RNA starts evolving more efficiently. You can have, for example, mobile elements and viruses, and eventually evolution. So basically, again, you don't have to think of a cell as it is today for evolution to occur. You can have sort of, some other forms of compartmentalization. These are thought to occur from, for example, bubbles in, geological formations that undergo evaporation and, these sort of shallow seas. Or they're also thought to possibly occur in the deep ocean in these hydrothermal vents, these, you know, black smokers initially, and more recently we think that the white smokers might be the reason for that, where basically you have this influx of energy from the soil that basically creates these self-replicating segments. They don't have to be fully autonomous. They just need to be self-replicating or cross-replicating. So these are still what you see today in mobile elements, in viruses, and sort of eventually you kick off evolution. You then can evolve cell splicing with groups 1, 2, and 3 introns. You then can evolve general splicing, the spliceosome that's still an RNA machine. Eventually, RNA decides that it's tired of doing all the work, and it's like, oh, listen, I'm going to make protein. It's a very different book of Genesis I'm giving you here, where basically RNA, all-powerful, says on the 7th day, I'm just going to rest. I'm just going to have protein do all the work. So basically, RNA eventually invents protein. You have translation, and that's sort of the ribosomal RNA, the rRNA that we still see today. And you now have RNA and proteins together existing. And you will notice that the enzymes that carry out reverse transcription are in fact proteins. The enzymes that carry out transcription, like RNA polymerase, are proteins. They're not RNA anymore, because frankly, RNA at that point was tired and it had invented proteins, and it's like, you guys do some of the work. So the other thing is that we call it transcription to go from DNA to RNA, and reverse transcription to go from RNA to DNA. But of course, reverse transcription was invented first because in RNA world, there was no DNA. So you had to first, quote unquote, reverse transcribe it, and then eventually invent transcription from DNA to RNA. And then of course, RNA, protein, and DNA eventually specialized, with DNA becoming the boring, super safe, super beautiful molecule that you only keep inside your nucleus for information storage. Proteins becoming the super messy catalysis, sensing, structure, and, you know, all of the hard work that RNA was tired of doing, and sort of huge more potential for evolution given the 20 amino acid building blocks rather than just 4 letters. And then RNA, of course, still has vestigial functions that have not been fully replaced, and it can still carry out all of the above in different parts of life, where you can see its roles in information storage in some species, in catalysis, sensing, structure, etc., and so on and so forth. Okay, so let's see who's with me so far on sort of all these awesome functions of RNA and these beautiful, you know, world that existed fully out of RNA before our modern world with the central dogma, you know, became a thing. So very cool. So 158210, and then who feels that they've learned something today? So okay, so again, you can read a ton more about RNA world. It's a super, super cool period of our ancestors. So we have 144710. Okay, so now that I hopefully have convinced you how awesomely cool RNA is, so basically I talked to you about ancient sort of housekeeping non-coding RNAs and the RNA world hypothesis, which has been, you know, solid since the 1960s. So, you know, it's probably true. And then some young non-coding RNAs that have also more recently evolved, and these include additional regulatory layers in complex organisms. For example, microRNAs were only recently evolved. Long non-coding RNAs were only recently evolved. So the, you know, the RNA world is not some just vestigial thing of the past. It's also a layer of evolution that, a layer of regulation that evolution will explore. So anyway, so RNA is totally awesomely cool. So let's now switch to how do we infer RNAs from the genome, and then how do we fold RNAs into all those super cool structures. So in the previous lecture, we basically saw how we can treat gene expression analysis, where we basically take the mRNA levels as a single value with a vector of expression for every gene in a bunch of conditions, And then as you build all those conditions together, you basically build your matrix of expression. And then you can do all kinds of things like gene expression clustering, gene expression classification, discovering functional classes based on their clustering or their features, and so on and so forth. Today, we're going to dive into what gave us these values in the first place. So how do we reconstruct transcripts? So we're going to look at how modern methods that basically start with RNA sequencing can infer gene expression levels, and then how to, number one, reconstruct transcripts, how to reconstruct transcripts even without a genome, and then how to predict differential expression using a lot more information than simply the the number of reads. So this is basically what we talked about last time. We basically said, oh, microarrays hybridize directly, and then RNA-seq sort of counts RNA molecules. So let's now dive into this part and see how does RNA count these molecules. So the basic premise is that in data generation, you basically start with mRNA or with total RNA, and then you remove any kind of contaminant DNA. You sort of remove all of this. And then you also can select away a ribosomal RNA or select for polyadenylation, which is usually found at the end of RNA molecules. And then you end up with a more purified set of the RNAs that you care to study, usually mRNAs, messenger RNAs. You then reverse transcribe those into cDNA. And you could also use strand-specific RNA-seq or double-stranded RNA-seq where you don't know which strand was the original one. You can ligate adapters and then PCR amplify potentially with single-cell RNA sequencing. Sometimes you avoid PCR amplification because it can lead to all kinds of biases, but with most traditional approaches, you amplify. And then you can select a range of sizes, and then you sequence the ends of these molecules. Why do you sequence the end of the molecules? Because sequencing long RNAs and long DNA in general is difficult. The reason for that is that a lot of our sequencing reactions are, well, traditionally, they were based on the ability to distinguish between a very long transcript and a transcript that's sort of one base longer. So if I had, a length of, 354 nucleotides, I could tell that that was ending in an A. And then if I could tell that length apart from something that's actually 355 nucleotides, I could then tell that that ends in a G, okay? And then the reason why traditional sequencing technologies could go only up to, 900 or 1,000 bases is because eventually I can't tell the difference between this length and that length. And the way that I could tell the difference is by sort of, in the olden days, by having, oh, Perez, you're unmuted. In the olden days, I could basically have, 4 different wells, one with reactions that involve A, one with reactions that involve G, one with reactions that involve C, and one with reactions that involve T. And I would basically grow my RNA, my DNA, by one base at a time, and then see which of these tubes would light up. So basically in sequencing DNA, what I ended up with is, sequencing reactions that look like this, that basically said, oh, something lit up here, something lit up there, something lit up there, something lit up there again. And then as long as I could tell the nucleotides apart from each other, based on their lengths, I could effectively read this by saying, oh, this is clearly a sequence that goes A, G, T, T, C, C, G, A, C, T, C. So that's the old traditional reactions. It's a little more complicated because some wells could recognize either C or T and so on and so forth. And the way that this was possible is by a series of reactions that would basically first incorporate, all faulty nucleotides that could only grow by one, and then wash them away, and then incorporate fine nucleotides that can grow by one more, and then so on and so forth. So basically, you would allow the reaction all the way to the incorporation of one more base, and then stop the reaction. One more base, stop the reaction. Then you could see, you know, initially radioactively, and then, you know, subsequently fluorescently, what was the nucleotide that was inserted. Okay, so this is the very old technology for sequencing. So let's see who's with me so far. Awesome. So basically that's, okay, so 28001. So that's the old sequencing technology. The more recent sequencing technologies are instead, and basically this technology was eventually replaced with, you know, hugely parallel capillaries that would sort of have, you know, 384 well plates where you would basically carry out 384 sequencing reactions simultaneously. And that was really awesome because you could sort of sequence a lot more than when sort of a grad student would sequence one of those a day. And, you know, these 384 well plates gave rise to sort of more and more automation. But then at some point, next generation sequencing became possible. And what next generation sequencing does is that it basically creates sequencing reactions in a huge field of RNA or DNA molecules that are sort of, you know, plated away without necessarily having a coordinate system. And now every one of those reactions is effectively creating an you know, DNA molecule simultaneously, one nucleotide at a time. So with imaging, you basically now have the ability to sort of look at this in super, super high resolution and eventually see as the different colors get incorporated. So you can tell when an A was incorporated, you know, in this reaction, the A was here and here and here and here. And then in the next cycle, oh, there was a G, you know, here and here and here and here. And in the next cycle, there was a T here and here and here and here. Okay, so eventually you would basically, from this visualization, from this imaging, you would basically be able to tell all of the sequencing reactions that have happened at every point in your huge field, you know, hundreds of thousands of molecules at a time, rather than only 384 molecules at a time. Okay, so who's with me now with next generation sequencing? So basically the whole concept is that instead of having wells anymore and all of this capillary based automation, you now have plates, next generation imaging, super, super high density sensors, and then growing molecules simultaneously across this whole field and being able to sort of image them directly. So 174310, this is awesome. So that basically means that with the old sequencing technologies, I could only go to, I don't know, 1,000 nucleotides. But with the new sequencing technologies, I can actually only go to, I don't know, 70 nucleotides if I'm lucky. So that's a problem. This is hugely cheaper, but much, much shorter. So, you know, this used to cost $1 a nucleotide to sequence the entire human genome. And this basically costs $2,000 for, you know, about 3 million nucleotides. So, you know, sort of basically six orders of magnitude cheaper. It's basically, you know, a tremendous sort of drop in cost. All right, so you know, this is all nice and good. So we can now sequence DNA massively parallelly, and that's sort of what has led to this whole concept of, hey, instead of just using microarrays, let's use sequencing. But that also means that we can only sequence small segments at a time. And the way that we do that is that because we can only sequence, I don't know, 30 to 70 nucleotides, we basically only sequence the beginning of the molecule, and we also sequence the end of the molecule, how can we do the beginning and the end? By having different primers that basically, hybridize and start the PCR reaction from this way or from that way. And then in the end, we know based on size selection of the fragments here, roughly what their distances will be. And then we can kind of infer roughly where they are on the genome. So basically the computational challenge ahead of us is how do we go from sequencing reads to the transcriptome, okay? So let's see who's with me so far in sort of understanding the premise of the way that we're going to be sequencing RNA is by converting it into these short DNA segments after fragmentation and then site selection. And then we are only able to sequence the tail ends of these here. So the question is, why are we only able to sequence 70 base pairs with next generation sequencing? The reason for that is that let me end the polling here. So 1510-000, that's great. If you go back to this, eventually these molecules kind of fall apart. You're not able to sort of sequence them all in the same density anymore. And then, these reactions just become very, very error prone. So there are sort of long, molecule RNA sequencing reactions that are now, DNA sequencing reactions that are now becoming commonplace, where you basically sort of have, for example, with nanopore, you have one strand of DNA that's sort of pushed through a tiny, tiny little nanopore. And as the particular letters are going through, you can kind of see the, the signal of that DNA molecule. Like basically when it's an A, it has a particular shape, and then you can sense that. And when it's a, a T, it has a different shape, and then you can sense that. And when it's a G, it's got another particular shape, and you can sense that as well, and so on and so forth. So basically, you can sort of flow a single DNA strand of DNA through one of those pores, and then measure the electrical properties as it's going through. That will sort of tell you, oh, this is sort of the sequence of nucleotides. And there's more and more technological advances that allow you to now do this in sort of longer and longer fragments with smaller and smaller error, and so on and so forth. Okay. So I'll go back to the other one. Okay, so that's now the computational challenge. How do we go from those reads to the transcriptome? So the very simplistic way of seeing this is you start with a sea of RNA-seq reads, and you can either map each of those reads to the genome and then assemble the transcripts from the spliced alignments, or you can assemble these de novo to each other and then build the transcripts and then align the transcripts to the genome. So basically, in one approach, the genome in blue here comes first, where I map all of my reads to the genome and I use that as my scaffold. But in some cases, I don't have a genome. And then, for exotic species, for example, where you only have RNA, and then I can just simply assemble these reads to each other de novo and then align the transcripts to the genome. Okay. So now let's look at these map-to-genome methods first, and then these Align-de-novo methods second. So the map-to-genome methods basically start with first an alignment of unspliced reads to the genome. And at the core of this is a super, super fast short read alignment using the Burrows-Wheeler transform. Okay, so that's called BWT for short. And then the program Bowtie is sort of reminding you of this BWT. And then that was the first program that was basically used for mapping these short reads very, very fast. And we're going to talk about how Bowtie works. So Ben Langley developed that in Johns Hopkins in collaboration with Steven Salzberg, and later with Leo Pachter. And then Cole Trapnell was initially working with both of them as well, and then sort of developed Top Hat, and then Loyal Goff in my group and in John Wren's group developed Cummerbund, and then a collaboration of other characters basically contributed to this tuxedo suite. It all started with BWT, and then it became sort of different names with cufflinks, where you're sort of putting together the links, top hat that sort of sits on top of the bow tie, and so on and so forth. So there's all kinds of cute names associated with that. But basically, the whole pipeline is that number one, you first align the unspliced reads to the genome using super, super fast alignment. You then have some candidate exon centers and some potential splice sites, okay? So the idea here is that many of your reads will align contiguously. So you don't have to worry about the spliced reads yet. You align the contiguous reads first, and then after you've aligned all the contiguous reads, you then say, oh, well, wait a minute. The beginning of the sequencing read maps to one place of the genome, and the end of the sequencing read maps to another place of the genome. How can that be? The way that this can be is because this region probably corresponds to a splice site, where basically, as the, this is an exon, and then it gets spliced to that other exon, and then that's the end of another exon, and so on and so forth. So you can kind of infer the exon boundaries based on the fact that there are multiple reads that are aligning at the same position, okay, and that are sort of stopping their alignment at the same position. So that's sort of the second part. That's where top hat comes in, where you want to align spliced short reads using particularly marked splice sites. You then want to reconstruct the transcripts, and that's where cufflinks comes in, where you link these spliced short reads into transcript alignments. And then what CuffDiff was doing in Cumberbund is basically differential expression analysis with probabilistic assignment of the read counts to the transcripts, and eventually visualization and analysis to basically ask and address biological questions using all of that. Okay? So who's with me so far on sort of the rough organization of this pipeline? So, first you align the unspliced reads, then the spliced reads, then you mark the splice sites, and then you sort of construct the transcripts, you figure out the differential expression, and then you analyze the results. Awesome, so 129300. All right, so that's basically a huge, awesome set of characters, and that's sort of the overall combination of tools. And let's look just a little bit in more detail, the step one of mapping the reads to the genome. So this is the Map-to-Genome method. So the whole idea is that you're going to be mapping the reads to the whole genome with bow tie. You're going to be assembling the consensus of the covered regions, and then you're going to be generating possible splice sites between neighboring exons. And then you're going to collect all of the initially unmappable reads, and then build a seed table from those unmappable reads, and then map the reads to the possible splices via seed and extend of each of the reads based on that. The Burrs-Wheeler transform itself is totally awesomely cool. It's way, way faster than hashing because it doesn't need to build a very big table in the genome. And we're going to talk about this Burrs-Wheeler transformation for initially used for compression in the epigenomics lecture when we talk about short read mapping again. But suffice it to say that it's a very memory, low memory footprint method that works as fast as hashing, but in practice much, much faster because you don't have to sort of have this giant table in memory. So the second step is mapping to the genome and dealing with the splice reads. So how do you align the RNA-seq reads to the genome? So the problem is that bow tie and other general purpose aligners don't allow large gaps in the alignments. So basically what you want to do is have your two ends of your, sort of molecule. First, you can find pairs where one of the two has already aligned uniquely, and then the other one must be somewhere nearby, and then you can extend it until you find that splice site and then extend further. Or you can basically use the known splice sites as markers for potential splits and then infer the splice read alignments that way. So basically the alignments for the pieces are stitched back together to make the full read alignment. And then that allows you to create the transcriptome. And then the last step is resolving individual transcripts and their expression levels. So basically we are now talking about first the individual reads and where they go. And now we want to actually construct the actual transcripts. And that's kind of challenging because you basically have to infer the expression level of every transcript as you're mapping the reads in order to, in some cases, probabilistically assign the reads to the right transcripts. So yes, indeed, the gene expression levels will be proportional to the number of reads. So genes that have more reads will basically be inferred to have more expression. But you need to correct for the total gene length. For example, a very short transcript will only have, I don't know, 30 reads, and a long transcript will have, 300 reads, because it simply was a longer transcript. There's more, opportunities for that transcript to be hit. So you want to normalize by the fact that, this was a longer transcript and therefore had more reads, more RNA molecule, basically more RNA nucleotides for the same number of RNA molecules. So basically, if there were 4 transcriptional events for this transcript and 4 transcriptional events for that transcript, this one will result in 30 reads and this one will result in 300 reads, even though there were only 4 events of transcription initiation. So you want to correct for that. You also want to somehow guess which splice variants every read is coming from. So basically the problem is you have to infer which transcript every fragment came from. And some fragments could have come from any transcript. So the black ones here fit with any transcript. Some only fit with the blue transcript or only with the yellow transcript. So basically there's no doubt that this one is part of this particular alternative splice form, and that this one is part of that alternative splice form. But this one is ambiguous. It could be part of blue and it could be part of something else as well. So, for example, this one could fit either red or blue because, it could fit either. So basically what you want to infer is the conditional probability that a fragment came from a given isoform. And you can infer that as a function of that isoform's existing abundance based on the other reads. So you basically have a large number of reads that are unique that allows you to now start estimating the isoform abundance for the ones that are unique, and then use these estimates to probabilistically assign the remaining ones to the transcripts that are most likely to have generated them. So you can basically have a likelihood function for every gene where you can take any set of reads and any set of transcripts and build a likelihood function that basically tells you what is the probability of observing the, a particular number of reads or a given read, given the transcript abundance. And using Bayes' rule, as we saw before, you can now basically say, well, what transcript abundance should I infer to maximize my likelihood function? So basically, we're going to be searching argmax over these transcript abundances in order to maximize the probability with which these transcript abundances are likely to have generated the reads that we observe. Okay. So we basically have, unique transcripts, uniquely mapping reads. And then when I'm mapping F1, I want to sort of assign F1 to the transcript that gives me the most parsimonious or the most likely interpretation of the data. Okay. So with the multiply map reads, you basically have a challenge because you don't exactly know where to map them. So you can basically use this expectation maximization approach to find the most likely assignment of every read to every transcript. And then this is what you use in the core of many of these methods like cufflinks, cuffdiff, RSCM, and then express. Okay. So that's the third part. We have to somehow guess which splice variant the reads are from. But you also need to correct for systematic biases in reverse transcription. Basically, because these transcripts are primed from the tail, the poly A tail, to the interior, when you look at the transcripts themselves, they're basically, sort of much more abundant. The reads are much more abundant at the end of the RNA molecule. So you have to walk back from that in order to sort of capture the remainder. So you basically model these sequence-specific biases where reverse transcription starts and stops, and then you can increase further the accuracy of your abundance estimates. Okay. So putting it all together, you basically have the last step, which is that you want to normalize the expression values based on the amount of sequencing that you did. Okay. So basically, the transcript map read count is normalized for, number one, the length of the transcript, and number two, the total depth of sequencing. And in the end, you report the number of RNA-seq as the number of fragments per kilobase, because you're correcting for the length, per kilobase of transcript, per total million fragments mapped, and then you're correcting for the total sequencing depth. Okay? And that's where this FPKM, so fragments per kilobase per million fragments, comes in. All right, so that's the Map-to-Genome method. You basically first map the individual reads, and you map the splice reads, then you infer the transcript abundance based on this expectation maximization model, and then you ultimately correct for the length, the biases along the transcript body, the biases with specific sequence patterns, and so on and so forth. Okay, so let's see who's with me so far. Very nice. So 50%, 20%, 25%, 0, 0. That's great. Okay, so that's the Map-to-Genome methods. It's also worth talking briefly about the Align-de-novo methods that basically, deal with the fact that perhaps we don't have a genome for that species. And therefore, you want to empower studies of non-model organisms and then infer the expressed gene content, the transcript abundance, and also differential expression for those. So the idea of these sort of assemble first, align later, is that you can basically generate a local genome assembly of all of your reads in the Bruin graph, which is basically a graph of consecutive k-mers that are different by one shift down the line. So basically, I don't know if you guys remember our initial discussion in lecture 2, I think, actually no, lecture 3, of the semi-numerical interpretation of string matching, where you basically compute the next character by shifting and adding and sort of deleting. So basically, you do the same thing here, and you construct a graph of all of the k-mers based on sort of how they match each other when you shift them by one. That allows you to sort of create graphs that have, splits and then remerges and then sort of deletions of introns and so on and so forth that allow you to eventually infer either sequencing errors or SNPs or splicing, and therefore maintain that ambiguity of your assembly according to each of the transcripts. Okay, and then eventually you collapse this deploying graph into just longer fragments that basically allow you to now reason about mistakes in the sequencing or genetic differences between the two alleles of a person, as well as splicing differences between two different transcripts from the same cell. And then once you've constructed this graph, you can now start resolving the graph and traversing it to basically infer the abundance of each transcript based on sort of all of the ways of traversing it. You can basically say, aha, there's a blue transcript that goes this way, there's a green transcript that goes that way, there's an orange transcript that goes this way, and so on and so forth. So that's the Align-de-novo approach, where you're basically first constructing this graph and then inferring these isoform abundances based on the total number of reads that sort of fit each of these paths. And then you partition that abundance according to the different alternative splice variants. Okay, so who's with me so far on the Align-de-novo and then Map-to-the-Genome methods? Okay, so we have 40%, 35%, 20%, 4%, and 0%. So, and then once you've done all that, you basically now have quantifications of the number of reads for every location. You'd like now to start inferring differential expressions between them. The advantage of de novo compared to mapping to the genome is that you can apply it to species that simply don't have a genome, or you could basically find transcripts from regions of the genome that are not very well covered in your assembly. You could basically say, well, maybe the centromeres are highly expressed and they're not very well covered in my genome. So I want to do a de novo assembly of those reads. You could also basically say, let me first do map to genome, find all the reads that map there, remove those, and then do a de novo for the remaining reads. And maybe I will find that there's all kinds of bacterial genomes and viral genomes and other sort of genomes that are coexisting in perhaps my human sample. All right, so diving into differential expression analysis, let's talk about how we can distinguish true differential expression from technical, biological, or stochastic variability. And that's where CovDiff comes in. You basically want to account for the variability in your fragment count for every gene across replicates and explicitly model it, and infer the fragment count for each isoform, along with a measure of uncertainty in this estimate arising from ambiguously mapped reads, which are extremely prevalent in alternative spliced transcriptomes. So the concept here is that instead of basically saying, oh, here's the value, now go figure out differential expression, you maintain that uncertainty throughout. And then the algorithm combines the estimates of the uncertainty and the cross-replicate variability under a beta-negative binomial model of fragment count variability, this is this, and then estimates the count variances for each transcript in each library. And then you can use those variance estimates directly in statistically testing whether two genes are differentially expressed based on the full uncertainty of your estimates. So the idea is that instead of saying, oh, here's the maximum likelihood estimate of the expression level, you basically say, well, there's a distribution of these estimates. And this distribution of estimates is basically allowing me to say, is this differentially expressed compared to that? So instead of basically saying, oh, here's the point estimate, here's the other point estimate, let me figure out their difference, I can basically say, what is the statistical difference between these two distributions and how likely are they to truly represent distinct gene expression level values. So who's with me so far with this whole concept of I maintain the uncertainty and then I use that uncertainty directly in estimating just how different are these distributions from each other. Okay, so 20%, 50%, 13, 13, 4. All right, so, and then once you've done that, you can basically either plot the differential expression as on the x-axis, you can plot the fold change, but on the y-axis, you can actually plot the probability, the confidence with which you have this fold change. So the confidence is basically given by the log 10 p-value of being wrong. And therefore, the higher, the more confident you are. And obviously, you want to sort of set thresholds that you can recognize something that is statistically significant, and possibly above a certain fold change. And if the fold change is too small, you simply never have confidence. That's why we call these volcano plots, because they're sort of high on the sides and very low in the middle. All right. So RNA-seq is basically a versatile method for transcriptome analysis, enabling quantification and novel transcript discovery. There's both genome-based and genome-free methods for transcript reconstruction. Expression quantification is based on sampling and counting reads derived from the transcripts. And then the fold changes that are based on very few read counts lack statistical significance. And there are many analysis frameworks for, both alternative and complementary approaches to support biological investigations. Okay, so let's see who's with me so far on the second part. So basically we talked about mapping to the genome first, and then sort of dealing with certain reads, spliced reads, and inferring the abundance, Align-de-novo based on these graphs and genome assembly, and then differential expression analysis by capturing that variability and then maintaining that uncertainty throughout. All right, so we have 32, 48, 12, 8, 0. Okay. So now let's switch gears altogether and basically talk about how we can go into constructing, now that we have our RNA molecules directly from our cell extract. We want to infer the folding patterns of those molecules. So basically we're going to talk about RNA folding first. How do we even represent RNA folding? And then we're going to talk about biophysical energy constraints for folding. Then we're going to talk about parsing approaches with context-free grammars, which are usually used for parsing text and sentences. And then these context-free grammars can be coupled with a probabilistic generation for every rule, for every production rule. And then that gives you stochastic context-free grammars. And then we're going to talk about how we can combine phylogenetic and biophysical information together, and then some advanced problems on kinetics, pseudonodes, RNA-RNA interactions, and so on and so forth. So this is different ways of thinking about your RNA molecule. So here's a tRNA, a transfer RNA, that is basically the sequence, the primary sequence, and then you can wrap that sequence around itself. So A, G, U, C, U, etc. A, G, U, C, U, etc. And then you have this looping of this GGG matching the CCC here. And then the second loop of, GUGUC, GUGUC matching GACAC, GACAC. And then sort of you basically have the pairing of this to that. And eventually this little segment here matches the end of ACUU, ACUU. Okay, so that's sort of the primary sequence, the secondary sequence, and then the tertiary sequence is the tertiary fold is based on the secondary fold by sort of using biophysical constraints. This is hard. This is basically using physics and simulations and so on and so forth. But this is, still in the genomic realm, so it's in many ways much simpler. So let's now focus on secondary structure of RNA, which in many ways dictates the tertiary structure, but is much easier to compute and is a sort of prerequisite for inferring the tertiary structure. So RNA secondary structure covers most of the free energy of the folding. So basically all of these base pairs capture most of the free energy, and then there's just a little bit of additional free energy that you can capture based on tertiary structure. It is a biochemically useful coarse grain of the 3D structure, and it is also an intermediate on the folding pathway to the full 3D structure. And it's often very well conserved in evolution, and it's computationally easier to handle. So that's why we're going to be focusing on the secondary structure of RNA. So there's many representations for the secondary structure. We talked about this, conventional plot that basically allows you to visualize it very easily. You can represent this also based on the pairing. If you start the sequence from here to there, you can start from here to there, and then plot exactly which letters are paired with each other. And you can see here that these lines are not crossing. And the reason for that is because we are not able to represent base pairing between here and say here. That would be sort of crossing these. For example, this loop here, if you bound that loop here, that's in the middle of the blue guys. So basically these nucleotides here would be mapping these nucleotides here, and it would have lines crossing. So we don't necessarily allow that, which basically makes it much more tractable computationally, but still captures the vast majority of structures. So that's one representation. The circle plot is a second representation where you basically taken this and stretched it out to become a circle. and then stretched out all of the links between them to become lines. The parenthesis representation also builds on the fact that there's no crossing, and therefore I can just go through and say, oh, start paren, start paren, start paren, start, 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 and then close, 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 start, 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 close, 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 start, 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 or sorry, close, 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 close. And that basically gives you this parenthesis representation underneath that allows you to, again, figure out what is base paired with what. A dot plot representation is very convenient because it allows you to see not only the primary fold, but also secondary folds. You can basically say, aha, this sequence here is self-similar with that sequence here. So maybe that's a ribose switch, maybe that's an alternative conformation. And then the mountain plot is basically the push-push and then pop-pop operations as you're sort of, walking through your sequence, okay? So who's with me so far on these different representations of RNA folding? All right, so 46, 46, 404. So the parenthesis representation can also be, mapped into this directly, by basically saying which, parts of your sequence are in fact matching which other parts of the sequence. And then in the dot representation, you're basically searching for these stretches of similarity between, this part here that matches that part there, and so on and so forth. So you basically have the sequence starting from A, C, A, etc., and itself. And then you're basically asking, in what places am I, self-complementary? All right, so now let's see, how can we actually fold RNA? So in many ways, what you're realizing is that this is not very different from our sequence alignment that we had before. So I can basically say, let me find local alignments that match between a sequence and its reverse complement, okay? So why don't we do that? Why don't we basically start building an algorithm similar to what we had seen previously in dynamic programming, using a scoring scheme that is uniquely suited to RNA. So basically, the question is, what do we want to optimize? And does dynamic programming actually apply? And what is that dynamic programming matrix? How do we define that recursion formula? And how do we traverse? In what order do we traverse? So that's where the Nusinov algorithm comes in. So basically, the objective function is to find the RNA structure with the maximum weighted number of nested pairings, okay? So if I want to sort of scan through the sequence and find how does it align to itself, I basically can choose to open a parenthesis here or not, open a parenthesis here or not, open a parenthesis here or not, close a parenthesis here, or maybe open a new parenthesis, and so on and so forth. So at every position, I basically am making a choice between whether I'm opening, closing, continuing, incorporating a nucleotide, inserting a gap, and so on and so forth. So I basically have an exponential number of possible paths or parses of my sequence, because at every one of those locations, I basically can make one of multiple choices. Maybe there's 4 choices, and that would be 4 to the n possible fold for a sequence of n characters. So that's an exponential space. We want to explore it in a polynomial fashion. So we're going to define a recursive computation using dynamic programming that allows us to do that. And the how is very, very cool and tricky. So it's similar to sequence alignment, but a little more fancy. So first of all, our scoring scheme, in order to find this optimal structure, is going to be the minimum free energy. And in a simple physical model, we can assign every pair an energy value. So remember, A can pair with U, and C can pair with G, and G can pair with C, and U can pair with A. So that diagonal is what has the minimum free energy. So if you have high energy, the molecule is floating around a lot, so there's a lot of energy there. But a low energy state is a state that sort of doesn't move too much and is well stabilized. So that's the diagonal here that basically captures the Watson-Crick AU, CG, CG, AU base pairs. Notice there are two elements that are off diagonal, but have also low free energy. And these are the GU base pairs that I mentioned earlier. So U can pair with G and G can pair with U, in addition to the traditional Watson-Crick base pairs. Okay, so that's sort of one complication, but there are additional complications. So the question is, does dynamic programming apply? And the answer is that the energy scores are themselves additive. And this is also the case with more complex energy models that sort of deal with stacking of RNA bases. So excluding pseudoknots, and the pseudoknots is what I described earlier about sort of the end of the loop, pairing with the other end of the loop that would create the split. But by excluding pseudoknots, we can basically split the problem into subproblems. And we're going to define this matrix Eij, which is going to be the minimum energy of the subsequence from I all the way to J. So basically, if I have my RNA molecule, it's going to be starting at I and then aligning all the way to J. What is that part there? Okay. And then the recursion formula is going to be moving from the inside to the outside. Okay. So let's continue. So here's the key idea. The key idea of the Nussenhoff algorithm is that we're going to be looking for these local stretches of similarity. So this thing here will basically tell me the score from the ith position to the jth position. This thing here will basically tell me the score from that position here to that position here. And that will tell me the score from that position here to that position here. Similarly, these things here correspond to the scores of these sub-pairings, and so on and so forth. Okay, so every one of these dots corresponds to the pairing of the entire subsequence. So what I'm going to be computing here is the score at every one of those boxes, the score of the maximum alignment, the maximum score alignment, the minimum free energy alignment that I can obtain with the entire subset of that sequence here. So what I'm going to be doing is using recursion to compute the score from the inside to the outside. So basically, as I'm deciding on sort of building this score here, I can basically say, well, that score is a function of the smaller scores that I have already computed with everything within my subsequence. Okay, so basically, I have, I have to sort of move from the inside to the outside. I have to move from the sort of center part here, where I'm sort of looping together I's and J's that are very close on the sequence, out towards bigger triangles, and then computing all of the scores at each of those positions, which I'm going to be using in my recursion formula. Okay, so let's see who can kind of see what we're getting at here. Basically, you don't have to get everything, but the ability to sort of see how this is creating larger structures as functions of smaller structures, I think is the key insight that I want you guys to get. So great, we have 32, 36, 20, 8, 4. So basically the initialization is along the diagonal, we're going to be sort of putting all zeros, and then we're going to be moving out from the diagonal. So basically, we're going to be starting from I between 2 and n, and then from I again between 1 and n minus L. So, and then J from the inside out for those. So basically, and at every position, we're going to be choosing the one of two options. Either, as I am sort of making the decision at that position in my alignment that corresponds to the maximum score between those two, I can basically make the position of perhaps I'm going to incorporate a nucleotide from both directions by extending both of them by 1. Okay, so this is what I'm doing here. So as I'm walking up here, I'm basically extending J. I'm moving J to the left, and I'm moving I to the right. So basically, I'm sort of extending this ij towards the outside and computing it as a function of whatever I was computing first, had computed first. So basically, as I'm extending this, score that I've computed here into the next score, one of the options is I'm just gonna sort of, choose the position that was immediately below me. So basically, as I'm computing ij, so basically, this is I plus, I don't know, 3, and then this is j minus 3, for example. And then this is I, and this is j. So to compute ij, I compute it recursively as a function of anything that's already been computed inside this triangle, okay? And that's the I plus 1, j minus 1, with whatever pairing score I have for xi and xj, and that's given by the table that I showed you on the previous slide. So that's one option. One option is to basically just extend both sequences by 1 and therefore choose the value that's right there. Another option is to basically break up my sequence into 2 parts. I can basically choose to break it up into 2 substructures. So one of them is going to be to the left after k characters, and the other one is going to be to the right after, k characters. Okay, so basically with the, basically the alternative, so every time I'm here, I can choose one of 2 options. I can either choose to extend both by 1, and therefore choose the value that was previously computed here, or because of the traversal order, which is going from the inside to the outside, I can basically choose to, split the sequence up after a certain number of characters k, and then nest around. Okay. So let's see who's with me so far. Basically, this whole idea is that we're now building a dynamic programming solution, building up from smaller parts, and every single time moving from the inside of the matrix to the outside of the matrix. All right, so we have 17, 35, 26, 17, 4. So quite spread out. Okay, so now the choices are whether I'm going to be extending by, both of them. And you can see here, that was our choice of moving straight diagonal. The other one is to sort of create a little bulge here by only inserting one nucleotide, creating another bulge by inserting another nucleotide, or splitting that structure into a bifurcation, as I was describing in the previous slide. So then as we're doing this, we can basically create the score moving from the inside to the outside. And then when I have the maximum score, I can sort of trace it back to the arrows for this. And every time I split into sort of 2 different substructures, I will have arrows that point me to here and that point me to there. And as I'm reconstructing my optimal solution, I'm going to be creating pairs of characters at a time. Okay? So the basic recursion formula is that, to compute ij, I'm going to compute it recursively, either by inserting one character or another character or both characters, or by splitting and then recursing into those 2. And then I'm going to be filling in my matrix from the inside towards the outside, and then traversing either, every row or, every column and so on and so forth. And then backtracking based on whatever option gave me the best choice to go down. All right, so that's the basic Nusinov energy folding with single base pairs. You can also build that with stacking between neighboring base pairs, because the stacking interactions between consecutive base pairs is actually sort of one of the most energy, sort of free energy capturing interactions. And you can also extend this to have interior loops, to have bulges, to have multi-loops between three different structures, to have, of course, stacked bases as we saw before, and then to have unpaired regions at the end of these hairpins. And then this is, using these pairs of nucleotides, because now you have all of these stacking energies, again, trying to minimize the free energy. And then you have the recursion, which is based on all kinds of options of what can happen for an I to J segment. I can extend by 1, or I can split into K. And then, when I split, I can have either a hairpin or an interior or 2. And, I can have this on the right, I can have this on the left, and I can have, all of these different options. So it's the same exact framework that you saw before, only now capturing these additional properties, okay? So that's for the maximum score, that's for the minimum free energy structure, but I can also infer a structural ensemble using the posterior probability. And that's basically using the same technique as we had talked about before of computing the total probability over all possible folds. And that's computed in the exact same way as we had seen before with dynamic programming, only now from the inside out. And again, that's called the inside algorithm or the outside algorithm score to compute the total score. And that's known as the partition function of the total probability of my sequence. Another very powerful way of solving RNA structures is through grammars, where you're basically inferring production rules that generate palindromes. So context-free grammars that you see in language parsing, more generally, is a series of sort of non-terminal symbols that can be translated into either other non-terminal symbols or terminal symbols, shown in lower cases. And these production rules are effectively dictating the type of sentences that your grammar can capture. And for RNA structures, the types of sentences are base pairs, for example, where you're producing 2 bases at the same time, or splits, where you're producing half a sequence, another half of a sequence. So you can basically build a grammar, a context-free grammar, for every RNA structure imaginable by having production rules that say I have my sentence, and that becomes AU or GC or GU base pairs, where I'm sort of generating them from the inside out, or a single base pair on one side or on the other side, or a split between 2 of them, okay? So who's with me so far on these grammars, being able to generate any kind of RNA structure by sort of using these production rules that sort of generate your sequences? That's awesome. And now, as I go through and quote unquote parse my grammar, my sentence according to my grammatical model, I can basically, okay, so we have 10, 43, 24, 10, 14. I can basically have this structure being generated as I'm sort of selecting these production rules. So for every one of those, I can basically have a probability associated with these production rules. So here, the production rules simply tell me what is the universe of sentences I can parse with my language. But if I have a probability associated with each of those, it becomes a stochastic context-free grammar that allows me to now infer the probability of a parse using the scores of the different production rules. And these basically allow me to now start scoring as I go through. And I can couple that not just with one sequence, but with a phylogenetic stochastic context-free grammar that allows me to walk up the tree and then generate parses that are phylogenetically consistent with each other as I build this sequence. Okay? So that's where we're going to stop for today. So we saw how awesomely cool the RNA world is, how we can reconstruct RNA transcripts, and how we can fold RNA transcripts using by physical energy constraints with a single base pair at a time, or with the stacking interactions energy model and how to infer the structural ensembles, and also how to use stochastic context-free grammars for parsing based on probabilities associated with production rules. So who feels that they've learned stuff today? Let's see. Beautiful. All right, so 43, 43, 13, 0, 0. All right, so that's where we'll stop today. And then on Thursday, we're going to be sort of switching gears and talking about epigenomics. And then on Friday, we're going to have another mentoring session, maybe, if we don't finish going through all the proposals today. And at 5 p.m., or I think 5:30 p.m. today, we have another mentoring session where we're going through, yes, 5:30 p.m. So TA Brian is basically giving office hours at 5, and then I'll be doing another session of feedback for your final project. So please come at 5:30 if you would like to talk about that. Sounds good? Cool. Thanks everyone. Bye-bye. Sorry for running over.
Thank you.